Okay. All right, welcome to Chapter 8. This is a no mercy tutoring session.
All right. What does he keep saying in class? No mercy.
No mercy.
No mercy class. I'm like, okay.
All right, so most of y'all heard this, but y'all's test is next Thursday, and we've got 3 chapters to go over, so I'm going to try to just go pretty quick today. Still, ask me questions if you have them. Participation is encouraged, as always, but I'm going to be going pretty quick, so let's go. No mercy. All right, Chapter 8, Carbohydrates. Why are carbohydrates important? Fuel for the body, right? That's like it's like the most easily accessed fuel that our body has. And so, as we'll learn on the next couple of pages, but like every cell in our body can utilize glucose in some form or fashion for an energy source. So it's kind of important that we have something providing us with glucose because it's obviously really important. So all living organisms use it. It's the most abundant class of biomolecules. If y'all never heard this, it's a carbohydrate, so it's a carbon that's got water on it pretty much. So every carbohydrate is really only made up of carbon, hydrogen, and oxygen. So that's why we see that CHO. So importance. Its role is structural and metabolic. So we have those. A lot of the carbohydrates are like the identifying cells or like the receptor cells on proteins, right? So what's the word I'm looking for? It's like a proteoglycans, right? Have you all heard of those? So like proteoprotein glycans, kind of like a glycogen. So those are really important for signaling. Like we just said, glucose is the most important carbohydrate. It's a major fuel. What else? And we can make a lot of other carbohydrates in our body. So glycogen is for storage, right? We know glucose is for use. Glycogen is for storage. Ribose for nucleic acid synthesis, because we have to have those 3 different things for a nucleic acid, right? We have to have the ribose sugar, the pentose group, and the what's the third one? Anybody? All right, we'll just keep going. We'll just keep going. Like I said, cell recognition, adhesion. It helps lubricate our joints actually, which is really cool. So having enough glucose in our body to make sure that our joints don't adhere to each other, and we can have the good movement, especially like in our synovial diarthrodial joints. Have y'all got to that part of DAA yet? Good, that's exciting. And it can have some structural elements. So like our there's a lot of like scaffolding that the carbohydrates actually help make in our body. Just help support it. Anyway, so important aspect here, classification of carbohydrates. Really divided into aldehydes and ketones. And so we'll see here at the bottom of the page and over the next couple of pages, the kind of classifying like a glucose is an aldehyde versus fructose is a ketone. A lot of other different examples that Dr. Sarkar is going to ask you about on the test. So knowing which sugar, whether it's an aldehyde or a ketone, right? Remember from chapter 1, differentiating the 2. Aldehyde is like a C with a double bond, O with an H at the end. So aldehyde is has a period, that H, whereas a ketone, it's really in the middle of the molecule. It's just the C with the double bond, O to it. So really, it's an aldehyde or a ketone. And then this is going to be a test question. Probably should put a little TQ here. That's not on. There we go. So knowing the different monosaccharide, disaccharide, etc. So mono, obviously, is just 1. So it has this carbohydrate just has 1 basic unit on it. Disaccharide has 2. Pretty simple. This is where people start getting tripped up. So in oligo, if we have like an oligarchy, right, we have multiple rulers. It's the same thing here. So oligosaccharides is from 3 to 6. It's made up of 3 to 6 monosaccharides. People usually forget what like the cutoff or the beginning point of that is. So they'll do like, oh, it's like 3 to 8 or like 3 to 5, what have you. So then a polysaccharide is anything that has 7 monosaccharides or more. So mono 1, di 2, oligo 3 to 6, poly more than 7. Really simple test question. So our monosaccharides, zoom in just a little bit more. Aldehyde or ketone derivatives that have a minimum of 3 carbon atoms. Not that important. But aldehydes are categorized as aldoses, which kind of makes sense. Aldehyde, aldoses. Whereas ketones are ketoses. Pretty simple. I think we can figure that out. And this is the general formula. So CH20 in parentheses times N. So it's the number of carbons that it has after that. Pretty simple. He won't ask you about that. So important here, though, is maybe subdivided into triose, tetrose, pentose, hexose, heptose, or octose. We're really going to stay up until hexose, which is really nice. So we don't have to worry about the 7 or 8 carbons of them. But we can see here we've got this nice little box. And y'all are all like, oh, what's in this box? Is it goodies? It's a lot of goodies. Halloween's coming up. This is the treat, not the trick. I'm really funny. So we've got, all right, so we've got our monosaccharides. So we're talking about monosaccharides only here. So just like one carbohydrate. And so all this triose, tetrose, pentose, or hexose is talking about the number of carbons within the monosaccharide. So a triose has 3 carbons. Tetrose, 4, pentose, 5, hexose, 6. We kind of talked about this a couple chapters ago. So that's just talking about how many carbons it has. Over here, we've got our aldose, ketose. Remember, aldose, aldehyde, ketose, ketone. Really, really easy way of remembering it is that if it ends in just OSE, it's probably an aldose. If it ends in ulose, so if it ends in ULOSE, it's probably a ketose. So it's a ketone or an aldehyde. So as we can see, aldoses, we have glycerose, erythrose, ribose, glucose, all ends in just OSE, right? So that's an easy way to say, hey, that's an aldehyde. Whereas we have our ketones minus dihydroxyacetone, we'll get back to that here in a second, but you've got erythrulose, ribulose, and fructose, but we'll have that down here in a second. So if you ever see just a ulose, automatically think ketone. All right, so glucose versus fructose. It's a really big differentiating thing, and we'll get to this when we start glycolysis here in a few minutes. But glucose, fructose, got to know those two. Glucose is an aldehyde, and fructose is a ketone. And I don't really have like a good way of remembering that as opposed to just remember it. Glucose is an aldehyde. So coming over here, so like how do we know which one is we have to know if like glucose is either an aldehyde or if it's a hexose. And this will also kind of come in when we get into glycolysis. But glucose and fructose are hexoses, so they have 6 carbons in them. All right, so the first enzyme of glycolysis is hexokinase, right? We talked about this last time. So hexose meaning 6, kinase, phosphate group. So we're adding a phosphate group to a sugar with 6 carbons on it, which happen to be glucose, right? So even just by the name of the enzymes, we can tell us what we do. So we already know glucose is a hexokinase, and glucose and fructose are pretty similar. And so we know that those are all kind of grouped together. Then it's really just remembering ribose and ribulose, right? Those are going to be along the same line. Erythrose, erythrulose, those are going to be on the same line. So another cool thing, which I think is on the next page. Yeah, these are, I moved it up. These are isomers of each other. So glucose and fructose, for example, they both have this equation C6H12O6. They both have 6 carbons, 6 hydrogens, and 6 oxygens in them. But they're just rearranged differently. You've got the same number. You've got 6 red Legos, 12 blue, 6 yellow, and you can build something one way, or you can take it apart, reuse those same 24 Legos, and build something else, right? So that's really all these are saying. So the aldehyde and ketone, they both have the same number of everything. They're just rearranged differently. One forms an aldehyde, one forms a ketone. Really simple. Main ones to know are, I would say, glucose, fructose, but the rest of the chapter, he kind of has it outlined as well for whether it's got 3, 4, 5, or 6 carbons. That's really just kind of taking 2 seconds or 5 minutes in between a class and just learning them. Questions? Am I going too fast? Is this okay? Cool. Okay. So structure of some monosaccharides. This is kind of just showing you difference between a ketone versus an aldehyde. So I guess going back over here, this glycerose can also be called glyceraldehyde. So it's pretty. We can kind of guess if that's an aldehyde or a ketone or not because it has it in the name. So we can see here glyceraldehyde. Do you think that's an aldehyde or a ketone? Aldehyde, right? Obviously. So if we know that dihydroxyacetone is like the opposite of that. So I just remember them by opposites. So if I see erythros, I know that the opposite is going to be erythrulose. Erythros is going to be the aldehyde. Erythrulose is going to be the ketone. So if I know that glyceraldehyde is the aldehyde, then I know that dihydroxyacetone is going to be the ketone. And you can see ketone, aldehyde, the groups are in different places.
You know, like put a structure on the test and ask what it is.
I don't think he'll actually put a structure. You'll have to know the name. He'll put the name on it. You want to name it from a drawing. Then we got glucose. Glucose has how many carbons in it? 6, yes. So that makes it a hexose, right? So is fructose also a hexose? Yes, all right, because they're isomers. So glucose is aldehyde or ketone? Aldehyde. So that makes fructose ketone. That's simple. And ribose, for whatever reason, whenever I see an R, I think 5. I don't know why. So I don't know. I might just be weird, but it kind of makes like 1, 2, 3, 4, 5. So that's kind of how I remember ribose is a pentose. That I don't know if y'all see that or not, but I see it. So that's all that matters. I'm joking. Y'all's information is way better if y'all learn it. So ribose is our aldehyde or ketone? Aldehyde, right? Because it just ends in OSE. So that would mean ribulose is a I guess that is not right. That's also an aldehyde because it's an aldo pentose. I thought that was ribulose at first glance. It definitely is not. So if you see anything that says aldo, it's going to be an aldehyde. It's going to be. If you see anything that says aldo, that's going to be an aldehyde. I'm sorry, I thought that was going to be ribulose, but following the format. But I guess it's different. I apologize. So stereoisomers. Got to know this definition. TQ. Got to know this definition. So compounds that have the same structural formula but differ in their spatial arrangement are known as stereoisomers. So they have all the same components, but they're just rearranged differently. So the presence of asymmetric carbon atoms allows the formation of isomers. So if he just talks about asymmetry of the carbon atoms, it's just isomer. But if he starts talking about they have the same formula but differ in their spatial arrangement, that spatial arrangement is what makes you think stereo, because I think stereo kind of talks about the shape in space. I don't know if any of y'all remember that from Gen Chem or Organic Chem, if you can back me on that, but I'm pretty sure that's right. Got to know those two definitions. And so this equation, I know he put this equation on our test as well when I took the class. So the number of possible isomers, so the number of different molecules that we can make, right? We can just rearrange it a certain number of way. What is it like if you have, like when you're doing probability and you're like rolling dice, you know, when you roll one die, you have a 1 in 6, right? But when you have 2 dice, you have to like multiply 6 times 6 to get that probability. And then like if you add a third dice, you got to like, so it's kind of that kind of logic where the more carbon atoms you have, you're kind of adding another variable to it. And so we're talking about the number of isomers that this can make. So glucose has 4 asymmetric carbon atoms. So you plug that into the equation, 2 up to the n, 2 to the nth power. So you do 2 to the 4, which would be 2, 4, 8, 16.
Are we supposed to memorize it as 4 asymmetric carbons?
So he'll tell you, so the test question would be something like, you've got glucose, you got a molecule of glucose, how many different isomers could that make? So you know that glucose, I think he would tell you how many asymmetric carbons it has.
Okay, that's on the math.
And you'd have to do the math.
Okay.
Yes. So I remembered, what did he do? I think he used the number 3 or 4 for us.
Will it be like an easy number, not like
It's not going to be like you've got like 23. No, he's not. No, it's going to be pretty simple. Number should be less than 1,000. But really, it's just the equation 2 to the n. If you remember the equation 2 to the n, you plug in the number that he gave you, make sure you multiply it by the right amount, and you're okay. Don't just double it. That's one of the biggest things that people do. They just double the answer. Sometimes that'll be wrong, especially if the number is like a 3 or something, as opposed to, yeah. So it kind of works for 2s, but if it's like a 3,
But you always multiplying 2 times, like it's 2 to the 4th power, so that always means you're multiplying 2s. So like 2 to the 4th is 2 times 2 times 2 times 2. It's 16. And 2 to the 3 is 2 times 2 times 2. Right?
So yeah, so I guess you can just keep doubling it then. Yeah, I'm confusing physiology. Yes, that one. Yes, don't just straight multiply it. Put it up to that nth power. Yes. Good. All right. So for example, if we've got ribose. Ribose has 3 asymmetric carbons. So that would be, how many isomers would that make?
oh, because it hasn't. No.
It's not in the. I just.
Oh, that was 8.
It would have 8, right? Because it's 2 times 2 times 2.
2 asymmetric ribose, look up above. D ribose, isn't that 2 asymmetric?
They might be 2.
Asymmetric.
He'll tell you how many. He'll give you the number,
And then you do math.
and you do the math. It's that simple. It's like, for example. All right. Sorry if that was confusing.
I just don't understand what asymmetric means, but he's giving us a number, it doesn't matter.
Yes, he'll give you the number.
Okay.
All right. So D or L isomers, glyceraldehyde. So glyceraldehyde, we know that glyceraldehyde is a triose. So it has 3 carbons.
What is that?
It's all. I think it's also called glycerose. But we know glyceraldehyde is a triose. So we can either have a D isomer or an L isomer. Like, what the heck does that mean? So D, the OH group, is just on the right, whereas L, that OH group, that hydroxyl group, is just on the left. That's all it talks about. So the next page, we have what the D and the L actually stand for. So D is dextrorotatory, L is levorotatory. And this was what I was talking about earlier on the chiral carbons, like the chiral molecules, so whether it rotates the light to the left or to the right. And so really what that's dependent on is this OH group. The main thing that we need to know about this is on the top of the next page where it says Only D isomers are used to synthesize carbohydrates in humans. He's got that in big, bold letters. Do you think that's important? Yes. So just know that humans, we all have D isomers for our carbohydrates.
Why do we even learn L isomers?
We won't really go into L isomers. He's just talking about, like, hey, here's our two umbrella terms. Like, other organisms will have L, but for the next couple pages, it's all just, you'll see that there's just a D in front of everything because that's all that we use as humans. And we're talking about our own biochemistry. So all monosaccharides are asymmetric except dihydroxyacetone. Don't think he'll ask you about that, but if there's like an all except for this one, he might pull that question. So just knowing that all are asymmetric except dihydroxyacetone, you can use as a fun fact for parties, know it for the test. If he doesn't use it, then you're okay. So optically active isomeric forms are known as enantiomers. So non-superimposable mirror images. So we got to know this definition as well. So we've got isomers, stereoisomers, enantiomers. So enantiomers are optically active isomeric forms. This took me a really long time to get through in undergrad. I'm sure there's a Khan Academy that, I think that's what I used as like to really make it better, was just a Khan Academy of those different three things. They'll say it way better than I will. But knowing those three definitions, it's going to be really important. There's a couple different ways that we can view these carbohydrates, these monosaccharides or disaccharides, what have you. The ball and stick model, where it looks like balls on sticks. The Fisher projection, or like a perspective formula. He's not going to ask you about like which. He's not going to ask you like, is this a Fisher projection or is this a ball and stick model or anything like that. This is just kind of general information. You're just showing like, hey, this is how you can write it if you were taking an organic chemistry class. But now to where the test questions are going to come from. He said you only need to know the boxed ones, correct? Yes. Good. All right, so these are all our aldolases. Aldehyde or ketone? Aldehyde, good. All right, so D, glyceraldehyde. Probably an aldehyde. That's got 3 carbons. We've got our 4 carbons. We just need to know erythrose. Right? All these are D, so I'm not just going to say the D because it's kind of implied at this point. So erythrose is 4 carbons. We've got 3 that are 5 carbons. We've got ribose, arabinose, and xylose. So kind of how I remember like the R's have 5, kind of like that X that I just made, kind of 2. So that X kind of has 5 points on it, 5 carbons. Really simple. So ribose, a kind of ribose looking word. So those are kind of similar. And xylose, 5 carbons. Question?
He asks us to name the groups. That's when we have to worry about the alpha, beta, gamma, or alpha, beta. You mentioned that in class. That if you're doing the numbers, you have to go by the top carbon on your way down, 1, 2, 3, 4, 5. But then if you have to use the Greek letter to name them, you don't use the groups on the top or the bottom. You just go with alpha, beta.
I don't quite understand the question. I'm sorry.
I remember saying that. It was really. You're gonna ask us.
Never mind.
Saying like if there's a change from like if you get 2 structures from ribose to a ribinose, the difference is in the, so if you were naming it, it would be the second, the second.
Yeah, so,
Second, but if you were using Greek letters, it would be alpha, because you don't use the top group.
okay. So alpha, beta, gamma, delta. Did you use the bottom group too or no?
Yeah, but if you only are counting using numbers, then you count the top group as 1, and then the next one's 2.
Okay. So it looks like y'all are starting from the aldehyde, right? From that aldehyde group.
Right.
And you're saying, hey, that carbon's part of that aldehyde group, so I'm not using that. So we're going kind of into the backbone of it and saying this first one after it is alpha, beta, gamma, because those are the first two letters of the Greek alphabet. If he said to know it, I would know it. If he says to know it, I would know it. So that's kind of how you can say, is like, oh, okay, well, D-ribose versus A-ribinose, they differ. They're structurally similar, except for the alpha carbon, the OH group has changed. I guess, I don't know. I don't remember him doing that specifically for us. But if he said to know it, absolutely know it. Maybe ask him in class tomorrow if he can give an example of how he might ask that on the test. He'll probably like that if you start off the day with a question. I don't know. Put him in a good mood for a hamburger.
Why does it call them like 3 and 4 and 5 carbons here instead of the triose and all that?
Why is it called?
Yeah, are those interchangeable or no? It's triose when you're talking about the entire like empirical formula.
You could put triose here. You could put tetros, pentose, or hexose here. Those are interchangeable, yes, absolutely. Great question. So, glyceraldehyde, 3, we know it's an aldehyde. Erythrose ends in just OSE, we know it's an aldehyde. That's 4. Ribose, A-ribinose, xylose, those are 5. They all end in just OSE aldehydes. Our 6 carbons, we have glucose, mannose, galactose, all end in OSE. Ooh, sorry. Yes. We have glucose, mannose, and galactose, all end in OSE. All right, we know glucose is an aldehyde. Glucose is an aldehyde. Glucose is an aldehyde. Say it again. That makes, yeah, oh, good. So we know fructose is what then? Fructose is a ketone. Good. All right. Galactose. All right, so now we have our ketoses. So we've got dihydroxyacetone, that's just going to be, it's not glyceraldehyde, so it's got to be dihydroxyacetone. We'll get to that here when we get into glycolysis. But dihydroxyacetone is our triose. I can't do that R. I have a speech impediment with R's actually. So erythrulose is our 4 carbon, right? Ends in ulose. We have our 5 carbon is ribulose or xylulose. Really simple.
There's no A-ribinose,
No, you won't have to worry about the A, ribulose. And then fructose is our 6 carbon. So really it's just the glucose fructose, which is a hard one. And then he might even just throw like a dihydroxyacetone just because it's a little different. And you may not be able to place that. But if you know that glyceraldehyde is the aldose, then you know that one's the ketose. So I kind of just compare. If y'all could just put it all on one page, just go kind of straight down the line and say, hey, this is ribose versus ribulose, like on the same level. And that's how I did it. But really simple. If it ends in ulose, it's a ketone. And just know if it's got 3, 4, 5, or 6 carbons. Epimers. So isomers differing as a result in the variation of the OH and H group on C atoms 2, 3, and or 4 of glucose. So how we saw earlier over here, we saw that all 3 of glucose, mannose, and galactose. All 3 carbohydrates that have 6 carbons. Yes? Yes. So they all have that same C6, H12, O6 molecules on them. So pretty much the only way those change is just how they're arranged, right? So starting with glucose here in the middle, we've got OH on the right, left, and then right. I guess starting from the top, he kind of starts on the top, so starts right, left, right, right. Well then, if you go over to mannose, we see here that the top 2 are on the left, the bottom 2 are on the right, and galactose is the third one switches over to the left. So really, it's got all the same component parts. They're just switched around. Really, just kind of spun around on an axis. And so the epimer specifically here is what he's talking about. is the difference between glucose, mannose, and galactose.
Just the right or left is the only difference.
The right or left of where that OH group is on the carbon, is on the structure overall, is the only difference.
Is it like knowing the definition or being able to look at?
He acted like he was gonna like put these on there.
Like the pictures on there?
Yeah, that's like.
I don't remember any pictures ever because it was all multiple choice.
For fructose, would you call it a stereoisomer or an isomer of glucose?
Don't ask me that question. Good question, though. Really good question. Would I call it an isomer or a stereoisomer? Is that what you just asked?
Yes.
I would say isomer. I would say isomer.
Your mic cord looks like a tassel coming off your little thing perfectly. Like, I was like, wow.
I planned that.
Well, you said it was an isomer?
I would say it's an isomer. Because based off that like picture, we're not talking about how it's arranged in space, right? It's just looking at kind of the chemical formula of it. C6H12O6 just arranged differently. But then when we get specifically into like, gets kind of confusing with epimer, where it's like, oh, it's just the differing arrangement. But that's only, the epimer is only taking into consideration the OH and the H group on carbons 2, 3, and 4. That's what makes it different. It's more specific.
Carbons 2, 3, and 4, you said?
Yes. How many more pages we got? Ooh, more than I thought. All right, keep them going. Glucose, the dominant monosaccharide. We can do the 3 different representations of it. We got a Fisher projection, Hayworth, chair. He might ask you like which of these is which. Chair, and I never got it, but it's supposed to look like a chair, and yeah, it's like a lounge chair. Yeah, it's a recliner. I don't know if turning it sideways helps at all, but chair formation, and then Fisher. I kind of think it looks like the Jesus fish, like the peace fish, except turned on the other end, so kind of like that, maybe not. And then just remember Hayworth. So we've got 2 cyclic forms of D-glucose. This is just kind of when he just starts going off on his own little tangents, I think. You've got alpha and beta, and really all it was is the difference in this OH group. Moving on. So D-fructose. Why is it D-fructose for us right now? Because it's in humans, right?
I thought D was to the right, too.
Yeah, D is also dextrorotatory. Good. So it's kind of the same thing.
Which one's which OH to the right? All of them? I mean, they all go to the right, but is it supposed to be a specific one or just?
Yeah, but
Yeah.
they're all to the right. Of the chiral carbon.
Oh.
Of the chiral carbon. So
Bunch of carbons.
I think you said the one on the bottom, like the
this one?
yeah.
Okay.
I have it circled for some reason.
Nice. So we're saying that this chiral carbon would have the OH on the right, but can't really see it. Just, yeah. Yeah. All right, so, sorry. Ribose, deoxyribose are used in nucleic acid synthesis. We learned this in previous chapters, right? We know that. We can just keep going forward. Sugars form glycosides with other compounds and with each other. So glycosides pretty much is just when they combine. So we can see here like glucose plus glucose yields maltose. We got to know that equation. So like maltose is composed of 2 glucoses. Going over here to the next page, sucrose is composed of, oh shoot, what was it? Sucrose is glucose and fructose, I believe. And then lactose is a galactose and glucose. So I'll say that one more time. So maltose is, so glucose plus glucose equals maltose. Glucose plus galactose equals lactose. And glucose plus fructose equals sucrose. So the glycoside would be just the combination of those two. I think the way he asked that was like, what two things form sucrose, or what two things form galactose? Polysaccharides. What does the prefix homo mean? Same, yeah. I guess kind of iso too. Iso would have made more sense, but homo polysaccharide, so it's made of the same things, whereas hetero is different, so made up of different monosaccharide units. Pretty simple. Glycogen, storage form of glucose. We've already talked about this. Really simple. I think we talked about this earlier too, but sorry, I'll get better at that eventually, maybe like week 12. So our starches are plant storage form of glucose, and those would be amylose or amylopectin. You might randomly ask that. All right, so this page, I think he's just showing us what glycogen looks like, where there's just a bunch of glucose units in succession adding to each other.
Up there, I remember that. 1, 4, whatever it is. I wrote a test question. You make like it's a linear chain, and then the one's like branched or something. I don't get it.
So, yeah, so I have that next. So amylose is a linear polymer of glucose units. So that's just kind of how we had that when we were making our proteins, our polypeptides. We just kind of had that chain for our primary structure. It's just that same thing. It's just a chain of glucose joined with alpha 1,4 linkages. You got to know those linkages. He's going to ask you about them. So alpha 1,4 linkage is for amylose linear. Amylopectin is a branched polymer of glucose. So that would be like up here, how there's like this linear chain, but there's also like a connection here, and it branches off like a tree, because trees branch. So you have amylopectin is the branched polymer of glucose, and those are joined with alpha 1,4 and alpha 1,6 linkages. So how I remember that is amylose, smaller word, so it's just alpha 1,4. Amylopectin is a longer word, so it's got both alpha 1,4 and alpha 1,6 linkages. And that's the alpha is just talking about the carbon, like what carbon and what position on the carbon. So amylose, straight, amylopectin, branched, or linear and branched. He's going to ask you, the answers are going to be like the alpha 1,4 or alpha 1,6. Those are probably going to be your answer choices. Other polysaccharides, cellulose has beta 1,4 linkages, whereas chitin has beta 1,4 and a homopolymer and acetyl D. Know it. Yeah, just know it. Just know that cellulose is beta 1,4. So we know that amylose, amylopectin, both A's, so alpha. Cellulose, chitin are both beta. So like the A's go together, and then B, C go together. That's helpful for me. Is that helpful for y'all? Okay. Going on. Those are plants, yes. So we've got amino sugars, so amino acids with sugars on them. So we've got glucosamine. This is just for y'all's benefit of knowledge. Have fun with it. Look over it yourself. We're going to keep moving on. Glycosaminoglycans. So these are components of our extracellular matrix. So we've got hyaluronic acid, chondroitin sulfate. He might just ask you what are two examples, or what's an example of a glycosaminoglycan. Just one of those two.
On the last question, sorry. It says something like be able to ID them. Amino sugars.
Be able to ID them.
Yeah.
What is he? Do you know what he meant by that?
What?
Do you know what he meant by be able to ID them?
Do you have that too? Okay.
Be able
What do
to.
we need to know about it? Yeah.
Be
able to ID. You told us that.
For the amino sugars.
Yes, I have the mean and the smooth.
Yeah.
On the amino sugars.
Can't understand. Give me a second. What is it?
On this page.
Be able to ID them.
Yeah, nothing.
Be able to feel its own
We did not have to
glucose and
be able to ID them, so this is new. All right, sorry, the microphone batteries died. So glycosaminoglycans, got to know the examples. Hyaluronic acid, chondroitin sulfate. And glycoconjugates, pretty much just like what can we put a sugar onto? We can put it onto a protein, we can put it on a fat, or we could put it on a different type of protein. Oligosaccharide linkages, got to know these as well. So sugars attached by O and N glycosidic linkages. So that's a test question. TQ, I already had it TQ'd. I'm going to double TQ it. So that'll be like, which of the following what type of linkages does a sugar do? And he'll be like, A is O, B is N, C is S, D will be A and B, and like C will be all the, E will be all the above. So you would put just D, right? Because it's only O and N. So you're probably going to throw like a third one in there to like, oh, I didn't study and I don't know what I'm talking about. So it's probably all of them, right? No. Just O and N. And that's the chapter. Questions? Sorry if that's kind of fast. 5-minute break. All right.
Are we going all the way to 7?
We're going to try to get through the rest of this chapter.
That's it.
That's it.
I thought.
The next chapter, chapter 9.
Oh.
All right, these. This is glycolysis. Gotta know these things like the back of your hand for this exam. So, how many ATP do we use? So, glycolysis is divided into a preparatory phase and a payoff phase. So, in the prep, we use a certain amount of ATP, whereas at the end, we make a certain amount. So we also need to know like the net. So we need to know the net ATP generated overall, right? We need to know where and how many NADH are made. A lot of that may not be quite familiar to you yet, but it will be. And we need to know what steps are irreversible. So just be thinking about these things. So like when I talk about them, you'll be like, oh yeah, that's one of the things that I for sure need to know. I'll tell you again, but these are the things we're looking at. So Dr. Sarkar, in these chapters, you got to know glycolysis, Krebs, electron transport chain, in and out. We need to know what the names of all the enzymes. There's only 10, so that's really nice. There'll be an 11th for like in between phase. And I think there's only like 9 for the citric acid cycle. So overall, just you got to know like 20 different enzymes. Not that bad. We need to know what you start with and what you make. So you start with something, you add an enzyme to it, you make something. You add an enzyme to that, you make something else. Add an enzyme, make something else. At the end, we'll end up with energy and we keep living. Really nice. So we get to learn about all this stuff. So you need to know all the names of the molecules. You need to know the enzymes that are being used. You need to know the order that they go in. So he could ask you, what is the order from like beginning to end? Are these like put these in the right order? And he'll just give you like 4 different options. If you don't know the way it works, then you're gonna get that question wrong. Lastly, oh shoot, what was it? What was I gonna say? It's not that bad. It's not that bad. So for people in front of you have gotten through this, people after you will get through this. From what I didn't study for this test as much as I should have, and I remember like learning a lot on this test, just because like when you haven't seen something, you're like kind of start putting things together. But for the people who did study, and they said they studied a lot, they walked out of that test saying that test was very straightforward. Either you knew it or you didn't. If you knew it, boom, circle the answer, next question. Boom, circle the answer, next question. If you didn't know it as much as you should have, kind of like how I did, I took that entire time and I was really uncertain on a lot of these different things. So if you can, watch this before the day before the test. Please learn this the day before the test, because if you have to learn both this, glycolysis, and the citric acid cycle the day before the test, there's going to be a lot of big words that's just going to like overwhelm you. If you're that kind of short-term memory person, good for you. You're the worst. But I'm not that kind of person. But if you after cells and tissues tomorrow, just sit down, learn 2 or 3 of them, or just rewatch this or rewatch one of Dr. Sarkar's lectures. It's going to be just okay. All right? Everything explains itself. Let's get started. Good? Cool. All right, so we got this one little carbohydrate page. Really all it's saying is that carbohydrates are important. All the cells in our body use them. There's 4 different distinct steps that we use to break down glucose in our body to eventually make this ATP, glycolysis. You make pyruvate into acetyl-CoA. Acetyl-CoA is important. Why? It connects all the different types of compounds, right? It can be acetyl-CoA can be made into a fat, a protein, or a carbohydrate, right? Test question, gotta know it was on y'all's quiz. It's gonna be on y'all's quiz that y'all are gonna take here, I think tomorrow, or if y'all had it Thursday. So glycolysis, making acetyl-CoA. Acetyl-CoA goes to the Krebs cycle, and then we get our respiratory chain with all of our ATP, the main ATP generator. TQ of these definitions, so gluconeogenesis is the creation of glucose from non-carbohydrate sources. So we've got a carbohydrate, and we're making it from non-carbohydrates. So glucose, new, genesis, creation of. All right. Glycogenesis is the synthesis of glycogen from glucose units for storage, right? Because glycogen is storage. So glycogen, genesis, the creation of, we're making glycogen. Glycogenolysis, so we see lysis. So when we see lysis, we're thinking breakdown, right? So the breakdown of glycogen to create or to give us glucose. So really differentiating between glycogenolysis and gluconeogenesis is kind of the one where people get mainly tripped up. Just because we're making glucose in both, but gluconeogenesis is from non-carbohydrate means, glycogenolysis is making glucose just by breaking down glycogen. This will be helpful later. The pentose phosphate pathway, just underline, where is it? Known as whatever, isn't that? It can also use glucose to make NADPH and pentose phosphates. Just if you ever see NADPH or a pentose phosphate, think pentose phosphate pathway, PPP. If you ever see PPP, NADPH, it's got a P in it, and a pentose phosphate. I think is like ribose 6-phosphate, but we'll get to there. All right, glycolysis. So glycolysis is the first place that we start producing energy in our body from sugars, right? So y'all have all heard like distance running versus like sprinting and how they kind of use different ways of making energy. So this is like the first one where it's like we need the energy now. We don't care if there's some like byproducts to it. I just need to run away from that lion, or I need to get to the cafeteria so I can be first in line. We're going to be doing mainly glycolysis. So glucose is really important, right? We've already said this, talked about it ad nauseum. We can use it to store energy, or we can make stuff with it, or we can put it in different pathways to do other things with it energy-wise, or we can just directly get energy from it. That's what that kind of top part's doing. So oxidizing it or taking off, so oxygen, we're taking off those hydrogens. So the oxidation of glucose is really important. We've got like a whole 3 chapters on it. So glycolysis is which glucose is oxidized to produce pyruvate aerobically. So that's a test question. That line I'll put it down here too. So the fact that we already know our end product, right? So we already know the end game. We're making pyruvate. That's going to be, we're starting with glucose, ending with pyruvate. So now whenever we see pyruvate, we know that's the last step. Done. But the big thing here too is aerobically. Aerobically. I didn't think that it was aerobic because at no point, anyway, I had this big long argument with him about like is glycolysis aerobic or anaerobic. I'm not even going to confuse you with like my argument of it, which is fine. It's an aerobic process. Uses oxygen. So, test question. It happens in what? I got that one right. I circled that answer begrudgingly. So anyway, so glycolysis happens in the cytosol, right? So we've already talked about we need to know where things happen at. What we talked about previously, like happened in the cytosol versus other places. Oh yeah, like the nuclear receptors versus like cytoplasmic receptors, membrane receptors, right? So kind of the same thing. we need to know where this is happening. So this is happening in the cytosol. The Krebs cycle and the oxidative phosphorylation happens inside of the mitochondria, not the powerhouse of the cell, because I hate that phrase. It's where we make energy within the cell, because no one knows what the powerhouse of the cell means when we say that. So this happens in the cytosol. Who can think of a reason why this would happen kind of in this big cytosolic area as opposed to within a organelle within the cell? Yes. So it can, so the whole cell, so the cytoplasm is pretty much everywhere in the cell, right? Yes. So it would make sense that if we needed quick energy, it would happen in the cytosol where it's readily available, as opposed to, say, one organelle needs energy or something else needs energy to do something. Well, we got to take it out of an organelle to get over there, but we need it fast. So that doesn't make a lot of sense. So glycolysis happens in the cytosol because it's just readily available to distribute it, the energy wherever it needs to go. It is made up of 10 enzymes. So glucose to pyruvate, and the energy released during the conversion of glucose to pyruvate. We make 2 ATP net, and we also make 1 NADH. So at the end of it, we're going to produce overall as a net. We're going to gain 2 ATP, and we're going to make 1 NADH. And you're like, what is NADH? Well, NADH, typically it's just NAD, but we oxidize glucose, right? So we took off that hydrogen. So this NADH is just a hydrogen carrier, and it'll take that hydrogen somewhere else to be used and made into energy later. That's all it is. I forgot what it actually stands for off the top of my head. So glucose transported in the cytosol. We use GLUT transporters. We'll learn that more in Try 2. I don't think he'll ask you about that, but in the cytosol. So we've got 2 phases of glycolysis. We've got the priming or prep phase, and we've got our payoff phase. So our priming, so it's kind of just saying, hey, like when you're making something in the kitchen, you prep it, you make it all ready, and the payoff is to have a food coma later, right? So kind of the same thing, minus the deliciousness that food could potentially have. So prep phase, we're going to use 2 ATP to make 4 ATP in the payoff phase. So people get confused here where it's oh, well, they're saying glycolysis makes 4 ATP, so why isn't it 4? Why did you say 2 earlier? Because it's a net of 2. So we use 2, negative 2, so we're down 2 already, right? We made 4, 2 puts it back to 0, then end up overall in the black with 2.
Produces 2 net.
Yes, 2 net.
And 1 NAD.
And 1 NADH, yes. It is 2 NADH. You are absolutely right. You are absolutely right. 2 NADH. So we have, good catch. So this is our prep phase. So pretty much it's split in half, 5 and 5 for the payoff phase. So prep phase is on this page. It's got a nice little heading. Payoff phase is on this page. Also has a nice little heading. So I like to use this box. To start off with. The second half, the rest of the chapter, kind of breaks it down into word form, and we'll probably do that after we go through this. I like to stick on this page first. So we already know step 1 from 2 chapters ago, right? So I can just skip that. No, I'm not going to do that to y'all. All right, so starting material. First thing to start off with, glucose, right? First thing, glucose. So our first step is we're going to turn glucose. We're going to take a phosphate off of ATP. So we're going to break ATP. We're going to hydrolyze it, right? And we're going to put that phosphate onto glucose, making glucose 6-phosphate. And the 6, like I said, the 6 is just the 6th carbon where the phosphate is on carbon number 6 of glucose. That's all it's saying. And we're using the enzyme hexokinase. Hexo meaning 6, kinase meaning phosphate, right? So we already know that glucose is a hexose, right? We just talked about that in the previous chapter. So it's a hexose. Is it an aldehyde or ketone? Aldehyde, aldehyde, aldehyde. So what this enzyme hexokinase is saying, hey, we've got a phosphate group, and we're going to put it on a 6-carbon molecule. So we go over here and we say, hey, look, here's glucose, 6-carbon molecule, and we're going to put a phosphate on it. It's that simple. It's just learning a language. It's just learning a language. All right. Irreversible reaction. Write that somewhere if you haven't already in your notes. Can't go backwards. So as we can see here, the line only goes one way. Right? On the next step, it goes both ways. So that would mean it's a reversible reaction that we can kind of undo it. But once we use this ATP, once we use that, we break it, can't go back. You took the green pill or the red pill or whatever. You can't go back. So got to know it. So that's where one of the first. So test question on here would be, which of the following is a irreversible reaction? And you'd say, bam, first one, ATP is used. What's next? So that's step 1. 10% done.
So anytime you use ATP, we can't reverse it.
You can't. So next step, glucose 6-phosphate to fructose 6-phosphate. Glucose and fructose are what to each other? Isomers, right? So look at this enzyme, phosphohexose isomerase, right? So phosphate, hexose. So it's a hexose, so it's got a 6-carbon molecule with a phosphate on it. And we're going to use an enzyme that makes an isomer. It's that simple. So we go over here, we're like, oh, glucose 6-phosphate, that's a 6-carbon molecule with a phosphate on it. And it's just going to turn it into a different 6-carbon molecule with a phosphate on it. And look, they're isomers to each other. So like that enzyme makes perfect sense. So fructose is an aldehyde or ketone? Ketone. That's got to be faster. Ketone. I expect more. So that's step 2. So it's just learning kind of the order. The order of these enzymes is going to be kind of difficult. You get like the wording kind of like switches up on each other a little bit, but still the enzyme describes exactly what the molecule is, which is really nice. So we've got hexokinase number 1, irreversible reaction, uses ATP. Enzyme number 2, phosphohexose isomerase. It makes an isomer. Glucose and fructose are isomers. Done. Step 3, phosphofructokinase 1, or PFK1. I'm going to keep saying phosphofructokinase 1, though. I found that helps a little bit more. Uses ATP. Do you think it's irreversible? Yes. So we know that steps 1 and 3 are irreversible because they're using ATP. He's not going to ask you like the numbers, like what number steps. He's going to ask you like what enzymes, but knowing the numbers, kind of like when we did the cranial nerves, you kind of like learn the numbers first, and that kind of helps you remember what they are. Excuse me, that's what we're going to be doing. So we're going from fructose 6-phosphate to fructose 1,6-bisphosphate. Oh, excuse me. So just looking at these molecules, we've got a phosphate on the 6th carbon of fructose. So going down here, we've got bisphosphate. So bis just means 2. It's like bi or di. I don't know why it's bis specifically, but whenever you see the prefix bis, it just means 2. So we can see there's 2 numbers here, right? So there's a phosphate on the 6th carbon, but we added one onto that 1st carbon as well. That's all we did. So going from step 2 to 3, all we've done is we've added a phosphate group. So if we've added a phosphate group, what kind of enzyme are we going to be using? What kind of enzyme? We're going to have a kinase, right? Because kinase are associated with phosphate groups. So look over here. This enzyme's got kinase in the name. That's super simple. And so what this is saying is, so we've got phosphofructokinase 1. The 1 at the end tells you, hey, look, I put a phosphate on the first carbon. We look over here. Oh, look, there's a 1 in the name. That's cool. And so we've got a phosphofructose. So we've taken a fructose with a phosphate on it, and we put another phosphate on the first carbon. Let's look over here. Oh, look, we've got a fructose with a phosphate on it, and we put another phosphate on it at the first carbon. It's that simple, y'all. It's that simple. Okay? Is this making sense?
Yes.
Good. Okay. So, irreversible reaction. Irreversible reaction. Only goes one way. Got to know it. So, our irreversible reactions up to this point are catalyzed by the enzymes hexokinase and phosphofructokinase 1. All right. So, he describes it as like these are kind of like priming reactions, and I agree with him. So whenever we kind of add a phosphate onto something, we're prepping it, we're priming it in a way. I'm trying to think of an analogy off the top of my head, and I don't have one. So maybe I'll come up with one in the next, whenever my brain works. So it's just getting it ready to do something. So this second priming reaction, we've got two. Let me start over. So the very first thing we do with glycolysis is we put a phosphate group on. That's the very first thing we do. So really that phosphate group is saying, hey, do something with me now. So glucose just by itself is not going to go through glycolysis. Once that first phosphate gets put on, that irreversible reaction of breaking ATP is what makes this whole series of events happen. So yeah, well, I mean, this is the first step of glycolysis, but like, you can.
So are those -2 ATP? Are those the two ones?
These are the first 2 ATP that we're using. Yeah.
So those, and then we add 4 and
We're going to make 4 in the second half.
So the net fraction.
So what I'm saying is here is that throughout the entire glycolysis, there's a phosphate group on every molecule that we've made until the end when we make those phosphate groups turn into ATP. So we can see here, first step, I mean, you can check it yourself. First step, it's got ATP on it. It's got a phosphate group on it. And then here at the end, there's still phosphoenol pyruvate, and at the end is when that last phosphate group is on. So really that phosphate group says, hey, let's go. Like, that's your buddy saying, get in the car, we're going downtown. So we can see here, so we've got, put the phosphate group on, we rearranged it, because whatever that wasn't working, we rearranged it, put another phosphate group on. So now we can see here that this step 4 with aldolase, we've got a phosphate group on kind of both sides of the molecule. So 1, 2, 3, 4, 5, 6 carbons. That's not a C, that's a C. Phosphate group, phosphate group. Yeah, so bis means 2. Bis equals 2. So what really we did, we just prepped it so we can say, hey, 6, like our whole goal with glycolysis and making energy is to break down glucose. Like we don't really care for glucose. We want all its, I'm that big business that buys up little businesses and just breaks them apart for the money. So what we did is say, hey, we put a phosphate on over here and rearranged it, put a phosphate over here. Let's break this down. So now we cut it in half. So now we've got 2 3-carbon molecules that are pretty much identical. And that's what our 4th step is. It's saying, hey, we're trying to break down glucose. And this is kind of the first big cut that we've got. I've got a big pie that my grandma made me, and I want to eat all of it. I got to make that cut. I can't eat it all at once, right? I got to cut it up a little bit. My grandma makes really good pecan pies. I'm sorry. So we can see here, like he's kind of got it highlighted different colors to kind of show you, hey, this is kind of where these cuts are going to happen. And then these next colors say, hey, this is what they look like afterwards. So we're taking fructose 1,6 bisphosphate, a fructose, just with 2 phosphate groups on them, one on the first carbon, one on the 6th carbon, and we're making glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both of them have phosphate groups on them. So kind of difficult words, but glyceraldehyde 3-phosphate, triose, tetrose, pentose, hexose. Triose, right? Because we just took a 6-carbon molecule and made 2 3-carbon molecules, right? So remember I said we kind of come back to this? So these are 2 3-carbon molecules. These are our trioses. One of them says aldehyde, so the other one's the ketone.
Well, why is that aldolase
Haven't figured that one out yet. I feel like it should be like aldoketolase, but it's only aldolase, so that's unfortunate. But the next step, triose phosphate isomerase, rearranges this one to make another aldolase, but we'll get to there in a second. So step 4, enzyme 4, we're cleaving the 6-carbon glucose and making 2 3-carbon sugars. The enzyme that does that is aldolase. So I guess how I remember it typically is we've gone from like this kind of bigger word, fructose 1,6 bisphosphate. It's kind of complex. And we're like, hey, that's too much for me. I can't take it. Let's simplify it. So let's just make it 2 3-1s, and we're going to have a really simple enzyme to do it, aldolase. So that's step 4. Questions? Do I need to start over from the top? Keep going. Keep going. Okay. So number 5. I'm going to weigh this down just a little bit.
This is 5 enzymes on the first page, but the 5 steps on the second.
Yeah, I think just because it's still that prep phase, we haven't made anything yet, so that's why he does it. And so he brings it in here, though. I don't know. So triose phosphate isomerase. Who can tell me what they think that means?
You have the 3 carbon groups, and you're going to add a phosphate and make isomer.
Very close. Very close. So if we added a phosphate, if we added a phosphate, what would that enzyme say? Kinase, right? We're not adding a phosphate. So this is a triose phosphate. It's saying, hey, we've got a 3-carbon molecule with a phosphate on it, and we're going to make an isomer of that. So all we're doing is saying, hey, we've got dihydroxyacetone phosphate. Like, I can't remember that word, so the body obviously isn't going to want to remember it. So we're going to turn it into glyceraldehyde 3-phosphate. So now we've got 2 glyceraldehyde 3-phosphates. Say again?
2 aldehydes now?
So now we're going to have 2 of those aldehydes.
No ketones.
So
No ketones.
These were already isomers of each other, so you're just taking the second isomer and changing the arrangement so that it is the same as the first. So
Exactly.
you need 2 of them.
Exactly. So it would make sense that the body said, hey, like, why would I have to, so I've got 2 of these things that are different. They're really similar, but they're different. So why would I have to come up with how, since enzymes are really specific, right? We learned, I've like drilled that into your heads, like enzymes are really specific to what they have. So why would I have to come up with like 2 sets of enzymes to get the end results when I could just say, hey, let's just change this one real quick, and then we can just use the same enzymes all the rest of the way down. That's why we're doing it. Question?
So if you see kinase, that's adding phosphate.
Yes. There might be one example later where it takes it away, but I don't think so.
Yep.
I think so, yeah.
Phosphoglycerate kinase.
So
Producing.
phosphoglycerate.
Producing.
Yeah. So
Same with the pyruvate kinase.
yeah. So like at the end, but I don't know, I guess you can see it like too. It's like it's making that as a product too. So that's where it kind of gets confusing. But like if you're introducing or taking away a phosphate, kinase. So like I originally just said, if you see phosphate adding kinase, I'm amending it to if you see it being added or subtracted to the equation, kinase. Still pretty simple. So that was our triose phosphate isomerase. We're taking a, where am I at? We're taking a dihydroxyacetone phosphate. We're saying you're not important. Sorry, body. and making it glyceraldehyde 3-phosphate. So now we have 2 glyceraldehyde 3-phosphates. So that's what these parentheses 2 are after each one of these steps. It's saying, hey, there's really 2 of these going on. So really we're making 2 NADH here, 2 ATP here, 2 ATP down there, because everything's just happening in parallel down each other. No, that's total. So yeah, so you could have like, I mean, if you didn't have like the parentheses 2 here, you have to like rewrite everything down on this side, each producing like one of these.
Because there's one on each side.
Yeah, so now there's just one going down this way, one going down this way. So he's just simplifying it, saying, hey, total 2 here.
So it's the same steps, there's nothing weird.
Yes. All right, so first enzyme is what? Hexokinase. What's important about hexokinase? Adding a yes. So more importantly, what is like what would be like a test question for that? What about ATP? It's irreversible, right? It's irreversible. It's only going one direction. So hexokinase, you're thinking irreversible reactions, the first step, gets everything going. So what's the next enzyme?
The isomerase, the
Phosphohexose isomerase. I'll put it up on the whatever so y'all can read it. Sound smart. We've got phosphohexose isomerase, right? So we're just taking, we went from glucose to glucose 6-phosphate, and we're going to change it into fructose 6-phosphate within isomerase because they're isomers, and they have a phosphate group on them. So next, we have what enzyme? Phosphofructokinase 1. Just get in the wording. It'll happen. It'll happen. If you want to simplify it to like PFK1, you can. How I remember that is like PFK1, like PFK, 3 letters, third step, whatever. What's important about that? Irreversible. Boom, bingo. Numbers 1 and 3. Hexokinase, phosphofructokinase 1, irreversible. Gotta know it, gotta know it, gotta know it.
Basically,
No mercy.
for the most part, kinase would be irreversible if you're adding phosphate.
Pretty much, yeah. Yes. Except
When you're not adding
there,
except when you're not.
in this setting, that would work 75% of the time. So it'd be a good guess if you completely forget. We'll get to there. So we've got hexokinase, phosphofructose isomerase, phosphofructokinase 1. So we added a phosphate to fructose 6-phosphate, so now we have fructose 1,6 bisphosphate. So that's way too big of a word. We need to simplify this. Like, let's just cut that in half. And that's exactly what happens. I'm so smart. Aldolase. So that's the enzyme that does that is aldolase. So that divides it into an aldehyde and a ketone, glyceraldehyde 3-phosphate, and dihydroxyacetone phosphate. Which one's the ketone?
Oh, the ketone second. Sorry, you got me.
Dihydroxyacetone phosphate is the ketone. All right, and like I said, the enzyme that does that is aldolase. Is that reversible or irreversible? Reversible. It goes both ways. The arrows say both ways. So what is the enzyme? So what's the next enzyme that we have? Isomerase. Triose phosphate isomerase. So if it's an isomerase, what's it going to do? It's going to make an isomer. So what are we going to start with and what are we going to end with? Yeah, so we're going to take our dihydroxyacetone phosphate and say, hey, nope, let's make glyceraldehyde 3-phosphate. So now we've got 2 glyceraldehyde 3-phosphates. And that's what we have. And so we can see here with the little parentheses 2 that we've got 2 of those. That's the entire prep phase. We've already learned how to glycolysis in like 20 minutes. Pretty good. Questions? Anybody? Now's your time to shine.
You don't need to know the structures at all, just like the concepts of what happens to them.
Yeah, you won't have to like, he's not going to draw like this little P, O, C, H, whatever. He's not going to do that, no. All words. Cool, so that's all the prep phase. How many ATP did we use in the prep phase? I need a lot louder, a lot more voices. 2. We used 2 ATP in the prep phase.
Step 1, step 3.
Step 1 and step 3, which actually happen to be our irreversible ones, right? Really simple. So now I would say more of the test questions are going to come from this side just because, look, like a lot more happens, right? Maybe like 60-40. But, so we've got 2 glyceraldehyde 3-phosphates. For all intents and purposes right now, I'm just going to pretend like it's 1 and just because I don't want to have to like count that many. Counting the 2 is too high. So we're just going to start with 1. So we've got a glyceraldehyde 3-phosphate. I'm tall, so I can touch the top of the thing here. So we're going to change that into 1,3-bis-phosphoglycerate. So if we started with glyceraldehyde 3-phosphate and we ended with 1,3-bis-phosphoglycerate, what did we do to it? We added a phosphate, right? Bis-phospho, 2 phosphates. If we see 2 numbers on it, we know that the enzyme before that added a phosphate to it. Yes, and so this one got added to that first position. And so this one isn't a kinase, so this is like might be a tricky. This one is glyceraldehyde 3-phosphate dehydrogenase. So what's more important here is not the fact that we have 1,3-bisphospho. What's more important here is we made this huge thing that's got a huge red circle on it, NADH, right? He's trying to make that stand out for a reason. So if we have a dehydrogenase enzyme, what does that mean? We're removing a hydrogen, right? Well, we're starting off with this NAD, and we're coming off with this NADH, right? So is it pretty safe to assume that that removed the hydrogen? Yes. That gets explained in a later chapter. It's actually, let's just do it. All right, we got time. All right. This is here. All right, NADH is an electron carrier. It's meant to carry electrons. So remember we kind of said that hydrogen's electrons can kind of be interchangeable, like we were talking about like transferring stuff. So how does this happen? So hydrogen, 1 electron, can be written as H plus electron. So if we have 2 hydrogens bound together, is that a, what is that? It's a polar or nonpolar covalent bond? 2 H's, nonpolar, right? Nonpolar covalent, they're the same. So nonpolar covalent bond be written like this. So we can ionize this or break it apart, and we can say that, hey, this hydrogen takes both of them. It's the jerk that takes all of the pie, instead of leaving some of my grandma's pie for me, and I get really mad. And then some who's left without. So really what that's saying is, hey, so this hydrogen is going to have a negative charge on it because there's 2 electrons and 1 proton, negative charge. And this one's got a positive charge because it's got no electrons, right? That proton in the middle is the only charge it has. So we got NAD has a positive charge, and the hydride ion has a negative charge. So when NAD plus is reduced, it produces NADH plus this H plus. So really that negative hydride ion, the one that has both the electrons, makes a bond with the positive charge over here, and we've got this leftover H over here. So that's why we get NADH plus H. So that's why whenever you see like the plus H, it's a thing. Don't worry about the plus H, just worry about the NADH part. Because that NADH is going to carry, it's an electron carrier, it's going to take it over to the electron transport chain. To the, well, yeah, so it says 2, so like because of the 2 parallel pathways. Sorry, huge tangent. Is that helpful? Did that answer the question? Okay. So glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. The important thing about the enzyme catalyzing this, so glyceraldehyde 3-phosphate dehydrogenase, this is one of the ones where, like, if you ever see dehydrogenase, like NADH or FADH2, something with an H on it is produced because we've taken a hydrogen off and we have made something else that's got that hydrogen on it.
Could that be reversible?
Yes, that's reversible because we're not using energy for that. It's just, we're not using ATP for that. So big thing. So test question. Which of the following enzymes produces NADH? You're going to look to the thing that says anything that's got a dehydrogenase on it, because that's what we're dealing with. So that's just the tricky one in like the wording, because we're like kind of adding a phosphate, but it's not a kinase. But that's less important to the production of the NADH. So this is the first time we made something. That's awesome, right? Look, yes, we're going somewhere, doing stuff. Paying off. Paying off. If only our student loans can do that too, but eventually. So now we're going from 1,3-bisphosphoglycerate. So that's a glycerate molecule, right, with 2 phosphates on it. So we're going from there, and we've got, hey, look, we've got 2 again. That's too many. So we're going to get rid of 1. We're going to get rid of 1, and that second one's going to make ATP. So anytime you see something with like 2, like a 1,3 or that 1,6 from the previous one, the next step, it's got to drop 1 in some form or fashion. So starting off with 1,3-bisphosphoglycerate, we're ending with 3-phosphoglycerate. So we went from 2 down to 1. And that, so we pretty much the ATP, the ADP took the 1 off of the phosphate off of the first carbon and made ATP. And that was phosphoglycerate kinase.
So I remember him saying that this stuff is reversible. Why?
He said that too. I don't know why. It would make sense. It has 1 arrow. I kind of want to say, this wasn't my book. I'm borrowing it from someone permanently.
When you drew it on the board, you just put a very small arrow because it's like 90% in one direction. But if it starts to go too quickly, then it'll slow itself down back regularly.
So the ones that are irreversible that he's going to ask you about on the test are 1, 3, and 10. Hexokinase, phosphofructokinase 1, and pyruvate kinase. Those 3 are going to be the irreversible ones. So the 3 irreversible reactions that he's going to ask you on the test are numbers 1, 3, and 10. So hexokinase, phosphofructokinase 1, and pyruvate kinase. Yeah, so it's that one. Yeah, so he had us rewrite that in. I guess he had to all rewrite this in, maybe not the arrow as big, but that is a reversible reaction to an extent. But, so we see kinase. So we're thinking phosphate did something, maybe ATP related. First step we make ATP with. Big deal. Do you think that's a test question? Is that specific enough? Big deal enough? Yes. Yes. So he can either ask, what's the significance of phosphoglycerate kinase, or what enzyme produces the first molecule of ATP, in glycolysis. Yeah, or he could ask you the going from 1,3-bisphosphoglycerate to 3-phosphoglycerate is important, why? So he could ask that from any one of those 3 points of view. It's all the same stuff, just kind of from a different viewpoint.
So the first molecule you're making it with is the 1,3-bisphosphoglycerate, right?
Say again?
It's not the bisphosphoglycerate, not the.
Yeah, so it'd be 1,3-bisphosphoglycerate to 3-phosphoglycerate. That reaction catalyzed by phosphoglycerate kinase produces ATP. And we have 2 ATP being made, right? Because we have those 2 parallel tracks going at the same time. So instead of writing both of those out and looking at them side by side, just easier and less ink to put a parentheses next to it. So we've made 2 ATP at this step. We're back to 0. We're square. We used 2 in the beginning in the prep phase. We just made 2. So everything from here on out is
Gains.
gains.
Hashtag gains.
Not hashtagging gains.
Hashtagging gains.
Hashtag gains. So, question. So this one just you see 1,3 bisphosphoglycerate, 2,3 phosphoglycerate. We're losing one of the phosphates, ending up with one phosphate at the 3rd carbon here. So something else, you have to know the order of it. So you got to be able to go hexokinase, phosphohexose isomerase, phosphofructokinase 1, aldolase, triose phosphate isomerase. You got to be able to do that in general. You got to be able to do that. The quicker, the better. But that doesn't really matter. That's the speed of it. So, he could ask you at any point, like, what's the order of these enzymes? So, or even if he just asks you specifically, like, what's the first one that makes ATP, you got to be able to place yourself, like you're like in that part of glycolysis, and say, okay, this is where I'm at. So if you're ever using energy, you're in that prep phase. You're in that first half of it. If you're ever making something, you're in that second half of it, because the first half doesn't make anything at all. One other thing is maybe I forgot all the words. Anything that has glucose, fructose, fructo is going to be in the beginning, right? Because that's what we're kind of starting off with. When we get to the middle, we're going to have more like glyceraldehyde, dihydroxyacetone, glycerate. And then at the end, the last 2, you can see both of the last 2 have pyruvate in it, right? Oh, sorry, you can't see that. So the last 2 have pyruvate. The one right before it is phosphoenol pyruvate. So if there's only 2 that have pyruvate, that's probably safe to assume that that's the one right before it. So just using these kind of context clues to put yourself in where everything's at, really, really, really helpful. So going back up, we are at 3 phosphoglycerate. So now we're at step 8. We're going from 3 phosphoglycerate to 2 phosphoglycerate. So all we're doing, we're taking that phosphate group, and we're taking it from the, where are we? The 3rd carbon, and we're putting it on the 2nd carbon. That's all we're doing. It's like, ooh, big shocker. Yeah, so it's mutating from 3 to 2. So it's not quite an isomerase, because all of the carbon enzymes are kind of still in that same chain, like all the carbons and oxygens are in the same kind of chain.
Isn't it the same thing? You're moving phosphate?
Huh?
You're just moving phosphate?
Yeah, you're just moving the phosphate. So that's why it's not an isomerase, because all the oxygens, everything stays the same except for the phosphate. So it's mutated. You got to go from high to low. So got to know that it goes from 3 to 2, because that's an easy thing that people get confused. They think it's like 2 phosphoglycerate, then to 3 phosphoglycerate. But if you look here, and you know that the step above it is 1,3 bisphospho to 3 phospho, then you know that the next one has to be 2, right? So this one, all we did was we took the phosphate group off that 1st carbon, leaving the 3rd carbon intact. So just using these kind of test-taking strategies, logic, and saying, hey, all right, it's 3, then 2, good to go. That enzyme was phosphoglycerate mutase. So we've got a phosphoglycerate, and we just changed it up a little bit, mutase. Really simple. 2 more. So 2 phosphoglycerate to phosphoenol pyruvate. This is an enolase. So you're making an enol. Don't ask me what an enol is. I think one of y'all did earlier, and it's just really difficult to explain. It's got it in the name. You're making an enolase. It's an enzyme with an enol. And so it's just releasing a water. So if it's releasing a water, what kind of reaction is that? Condensation, right? Condensation, right? We're making it rain. So condensation reaction. Really simple. So if you ever see pyruvate, or if you see phosphoenolpyruvate, and you're like, holy freaking crap, where the heck does that go? What's our end product? Pyruvate. So that's got to be the one that comes right before that. So then you can say insert, like phosphoenolpyruvate. Okay, what makes that? Well, it's not pyruvate kinase that makes pyruvate, which is the next step. It's enolase. So test question, what makes phosphoenolpyruvate? Enolase. Done. It's in the name. Don't make it harder than it has to be. It's the whole motto of this chapter. So last step. What do we do? What are we making here that's super big? ATP. So this is our last step of gains. Sure, why not? And so irreversible, right? So we just ended glycolysis. We made ATP. That's the whole goal of glycolysis, was to make ATP and to make pyruvate. Do we want to go back from that?
No.
No, we don't want to go back from that at all. We want to stay there. So that's an irreversible reaction. Irreversible reaction.
So 1, 3, and 10.
1, 3, and 10 are irreversible reactions. More importantly, the names of the enzymes, but still 1, 3, and 10 as a starter. Pyruvate kinase. Super simple, right? We're making an ATP, kind of deals with a phosphate. We're not making an NADH, right? We're making ATP, so kinase. That's glycolysis, y'all. It's not that bad. It's not that bad. So morals of story. We're gonna keep going. Let's just talk about it a little bit more. My voice isn't completely gone yet. So morals of story. Read, pay attention to what is, you're starting with something, you're ending with something. The enzyme has to describe that reaction. Just read it. If it says, phosphoglycerate, there's a glycerate with a phosphate, and then whatever else it says on it. So just context clues. It gives you the answers. You just pull out your decoder ring, which is your brain, use it, and you'll be just fine. Okay, so from the top, we've got glucose to glucose 6-phosphate. Hexokinase. It's cover it and quiz you. Okay.
Scooch it over a little bit.
No. You said quiz you. All right. Ribose, ribulose. We're going to go way back. Beginning, triose, tetros, pentos, hexos. Pentos, right? 5. So it's the R, kind of makes an X, 5 dots on it. Good. All right. So anything that just ends in OSE is a aldehyde. Anything that ends in ulose, ketone. All right. Besides the first one. And fructose. General rules. Good. I've accomplished my goal of getting short-term memory of that out of your head, so that's good. So first step. We're just going to go enzymes, just enzymes.
Okay.
So we've got first enzyme is hexokinase. What's important about it?
It's irreversible.
Irreversible. What happens? Why is it irreversible?
Phosphate, energy.
Because we're using energy, right? It's starting this whole thing. We don't want to. We want to start this reaction. We don't want it to end, right? So we don't want to take that back. No takesies, backsies for the first or last step. So second enzyme. So we've got hexokinase. What's our second enzyme?
Isomerase.
Yes, an isomerase. So phosphohexose isomerase. We just added a phosphate group to a hexose, so we went from glucose 6-phosphate. Now we want to make fructose 6-phosphate. They're isomers of each other, isomerase. Third enzyme. What does PFK stand for? Phosphofructokinase 1, right? That one's pretty important. He won't. I don't think he'll just put phosphofructokinase or phosphofructokinase 1 on the answer choice. So don't worry about that. What's important about phosphofructokinase 1? Irreversible. Why is it irreversible? Same reason as number 1, right? We were putting energy in. So we just put a second phosphate on. What do you think we're going to do next?
Split it.
It's going to split, right? We're going to take it off in some form or fashion. Too big of a word. What enzyme splits it? Aldolase, right? It was too big of a word, too big of a molecule. Let's simplify it. Aldolase. What enzyme? Do we make anything with aldolase? Like, do we make anything like energy-wise? No. Why not?
Because you're just splitting it right down the middle.
Yeah, we're still just splitting. We're still kind of setting it up for the payoff phase. What's our next enzyme? Triose phosphate isomerase, right? So we took a 6-carbon molecule, right? 6 carbons. If we split it in half, we've got 2 3-carbon molecules, right? So that means that triose, we've made 2 trioses, really. So it makes sense that that enzyme would describe the creation of those 2 trioses, right? Which one is the ketone?
The first one.
Oh, it's the second one. I say that every time.
Dihydroxyacetone phosphate, right? Because it's got that phosphate group on it. Each of them has a phosphate group on it. So what's the other one? Glyceraldehyde 3-phosphate. Cool. So we'll get, with studying, y'all get those. Also with your notes, you'll get those. So we've got a glyceraldehyde 3-phosphate and a dihydroxyacetone phosphate. Dihydroxyacetone phosphate is the ketone. It's also a triose. Cool. What are we doing next? We've got 2 of these things.
You're gonna add ADH, so it's the whatever.
Phospho something.
Dehydrated. No? Oh, I don't remember.
What's happening? Someone tell me what's happening.
Making the ketone into an aldehyde.
I thought we already did that with isomerase.
Well, yeah, that's what we did with isomerase already.
So now you add that in ADH.
I confused y'all a little bit.
Yeah, because you just talking about ketone.
Confused y'all a little bit. So, rewind. Aldolase splits it. Aldolase splits it. The isomerase makes dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. Is that clear? Did I confuse anybody with that? Okay. Everyone nod their head yes. Not peer pressuring though. That's bad. Okay, so we've got two glyceraldehyde 3-phosphates now. Yes? Yes. All right, those are happening. So the rest of these reactions are happening at the same time, pretty much. They're just separately, but we're kind of combining them for the next page. So next enzyme. Dehydrogenase. So if we have a dehydrogenase, what are we making? NADH, or potentially maybe an FADH later. But on this one, NADH. All right, did we ever make, y'all haven't had FADH yet, but did we make an FADH at all in glycolysis? No. When we all get to chapter 10, y'all are going to start making some of those. So knowing that glycolysis is only 2 NADH, no FADH, is important to know. Like, don't cross, it's going to be easy to like cross those over, so just understanding like what makes what doesn't make what, it's important. So, what's the actual enzyme's name besides dehydrogenase?
Oh, it's
I used to know these off the top of my head. It is glyceraldehyde 3-phosphate dehydrogenase. We were all so close, right? So close. I'll just keep it up. I'll stand in front of it. So, all right, so we made NADH. This is the first thing we've made. Step 6 is the first thing we made. Pretty awesome. So next enzyme, we have what? We're making ATP, right? So we just made an NADH. The next step is, hey, we've already made one thing. Let's make another, right? Let's keep the ball rolling. So phosphoglycerate kinase. We see kinase. Starting to think. ATP, phosphate group. Make ATP. How many ATP do we make in that step? 2, right? Because we have those 2 parallel things just going side by side. We've got those 2 glyceraldehyde 3-phosphates both going down. Next enzyme is what? Mutase, right? So we've got our phosphoglycerate mutase. So we're going from 3 to 2, right? 3 to 2. So now we've got 2. What are we going to make with enolase?
You're gonna put that word in there. Also, something enolase pyruvate.
Phosphoenolpyruvate, maybe?
Maybe.
Maybe? I want to confirm that. I think it should be, yeah. Phosphoenolpyruvate, right? So we see enol, it's in the name. This is enzyme 9, so it's probably going to have like a phosphate in the word somewhere because enzyme 10 makes pyruvate. So phosphoenolpyruvate. Do we make anything with this step?
No.
Do we?
ATP.
Oh, and water.
Water, right? Right? So kind of nitpicky. It's not like in bold or anything, but we make water in this step. 2 things of water, right? Because it's happening at the same time. So condensation reaction. We're making water. He could ask you, like, what type of reaction is catalyzed by enolase? See how it kind of carries over? Got to know it. Got to know the basics so you can apply it. Last step. What's important about the last step? Make an ATP. It's irreversible. We've achieved our goal of glycolysis. We've reached the finish line. What's our net? What do we get from it? 2 ATP, 2 pyruvate, 2 NADH, maybe 2 waters, but less important. It's less important. So 2 NADH we made, 2 ATP we made, 2 pyruvates we made. That pyruvate then gets taken and changed to, that's the next, yeah, it's the beginning of the next chapter. So, as we can see here, the next top of the page, we got glucose. This is just a really simplified version of everything. So glucose through glycolysis yields 2 pyruvate. All right, if only it was this simple, we only had to like learn it, just those words. All right, so test questions for sure here. For faux show. So in a hypoxic or anaerobic condition, really more like the fermentation. So if you ferment pyruvate or ferment ethanol in yeast, fermentation to ethanol in yeast. So if you have pyruvate and you ferment it, right, you make alcohol, right, kind of. You're going to get ethanol, right, because it's in alcohol. So fermentation of pyruvate yields ethanol in yeast. Whereas in a vigor, like when we're exercising in like a contracting muscle, we're going to make lactate. So that's more of an anaerobic condition as opposed to hypoxic. So hypoxic fermentates to ethanol. If we're exercising, it's going to be more lactate, right? Because we get that lactic acid buildup and our legs hurt and we like don't want to run anymore because we're like, oh my God, this hurts too much. And so we kind of stop and our coach yells at us. He's like, keep running. And we're like, no, I don't want to. Roughly. So got to know these test questions. So really the rest of this chapter just talks about each of these reactions specifically. Cofactors, know the cofactors for each one. It's typically magnesium. So we can see here, first one is magnesium, second one is magnesium, third one is magnesium. So when in doubt, we're going to guess magnesium, right? Magnesium again, magnesium again. Some don't use cofactors or coenzymes. So yeah, so like in glycolysis, if there's ever a cofactor, it's magnesium. Really simple. Enzyme is on the bottom, right? And so if we're either adding or taking something out of the equation, it's going to be happening on the top. So with this one, glucose, we're adding ATP using magnesium as a cofactor, making ADP, and that phosphate comes over here. It just breaks it down for you. It tells you everything I did that I told y'all. If it just helps you to have it broken down this way, I find it helpful to read over it again. Let's see, what else is there? So Remember when we talked about all those free energy changes in like a couple chapters ago? It was all like the delta G Gibbs free energy, right? So this kind of like puts it in perspective of like, hey, like this is how much everything costs to do. And we can see here like, okay, well, like this costs a little bit of energy. Like there's some require energy, some give off energy, what have you. But we can see here this last step, pretty big give off of energy, right? So also this step right here releases energy too. So right, so these were the ones that we made ATP with, correct? We should all be saying yes, right? So we made NADH here. We used ATP here. We used ATP here. So kind of bigger changes, right? So you won't ask any of these like specific number questions, but kind of putting into perspective, like, hey, we're releasing energy, we're using energy, what have you. Let's see, how much further do we have? So glycolysis can provide ATP in the absence of oxygen. So this is kind of what I was saying, where like glycolysis can be anaerobic, but for all intents and purposes, Dr. Sarkar's class, glycolysis is an aerobic pathway that happens in the where? Cytosol. Yes. So this is, like we said, overall net. We get 2 ATP per glucose molecule. Here's a big long equation if you care to like look through it. It's going to tell you everything we just already talked about. All right, mature red blood cells lack mitochondria. So this is kind of important, actually. So it relies on glycolysis as our source of energy. So what will happen after glycolysis is that we go into the mitochondria, right, powerhouse of the cell. Don't say it ever again, though, because no one, let's actually educate people, like, what the hell is a powerhouse of the cell? Sorry, my own personal tangents. And so if they don't have that mitochondria, they can only rely on glucose for their energy. They can only rely on glycolysis for their energy. So, you know, if you are diabetic or if you have sickle cell anemia, right, where your like red blood cells are changed, you can kind of see how your diet or your exercise or maintaining glucose levels or enzymes in your body can really have a major effect on how your body functions. So the next step, pyruvate is oxidized to produce acetyl-CoA. We'll talk about this next time, but gotta know. We'll just flip to it. Actually, we'll talk about it now. So, pyruvate. This is not part of glycolysis. Glycolysis ends with the production of pyruvate. But I'm pretty sure it's not technically the Krebs cycle either, so it's like this weird intermediate step. There's a test question on that somehow, and I like don't remember it. That's the one I got wrong.
You didn't have to give us like the names of each is like a certain vitamins, like COASH, like B5, the NAD is niacin, T2 is B1, FAD is vitamin.
Yes. On our test, he just kept it like this, I'm pretty sure. But definitely know the opposite. So that'll be more, I think it'll be try 2. We'll actually like learn all the B vitamins.
I feel like he really likes teaching us stuff from Try 2 already. He does that so often.
Yeah. Well, he teaches.
This next try, so why not just learn it now? And I was like, well,
If you can, do it. If you can do that, oh my gosh, that would be so helpful. Long-term memory, it's a like incredible thing. So we've got pyruvate. We ended with pyruvate, right? But now we want to make acetyl-CoA. Why do we want to make acetyl-CoA?
so they can be used by.
It's like super versatile, right? It can be used anywhere. And so kind of an important step. So this is pyruvate dehydrogenase complex. Gotta know that name. Gotta know all this crap that's on the top of it. So pyruvate dehydrogenase. If we see dehydrogenase, what are we thinking? Right, we're moving hydrogen, so there's probably going to be, look, there's NADH is here, right? Oh, look, it showed up. Some kind of hydrogen carrier is going to show up if you see dehydrogenase. If it's taken off a hydrogen, something's got to take that hydrogen somewhere else. NADH does that. NAD does that. So pyruvate to acetyl-CoA. We've got coenzyme A, or CoAsh, NAD plus, TPP lipoate FAD. Guaranteed 100% test question. Which of the following is or is not a enzyme or cofactor for this enzyme? 100% test question. There's 5 of them. So you're starting from pyruvate, to acetyl-CoA. So you know CoA's got to be in there somewhere, right? So CoAsh. Like CoAsh has got to be there because it's in the product, right? So if we know if it's dehydrogenase, like we know we're making an NADH, so we got to start with NAD, right? Because the H comes from pyruvate. And then it's just TPP lipoate FAD. Those are kind of like the oddballs out a little bit. But super, super important. We'll talk about that maybe at the beginning of next chapter again too.
Yeah.
Yes, maybe so multiple times being reinforced. And kind of here's that making lactate, making ethanol. Kind of running out of time here. Got to know that. Make sure you go over these parts.
For that one, why is it called a dehydrogenase when you're using hydrogen to make NAD?
Yeah.
Lactate dehydrogenase.
So like you're using the hydrogen, you're not releasing it.
So, no, you're not wrong. So, technically, you could say that your starting materials are pyruvate and NADH, right? You could say both of those are starting materials. And you're taking off a hydrogen and putting it onto something else. So in theory, like technically, we're removing a hydrogen, but yeah. But for our intents purposes, typically dehydrogenase for glycolysis and for Krebs cycle, you see dehydrogenase, you're making NADH. So this is kind of like a, not for those two, but it's still kind of similar, right? Because it's